everyone. So I'm glad to be here presenting our research to you. My name is Claudio Aguilar. I'm an associate professor in the Department of Biological Sciences at Purdue University. And our lab is interested in different things. In particular, we are interested in vesicle trafficking in general, and in particular in endocytosis. Specifically, we are very interested in how receptors interact with ligands, and the ligands then turn to activate the receptors by dimerization, for example, in the case of receptor tyrosine kinases, and that is followed by recruitment of different elements of the intracellular machinery. Some of them are required for post-translational modification of receptors, such as ubiquitination, phosphorylation, and others are required for the assembly of the endocytic machinery, for example. In this case, I'm showing you adaptor complexes like AP2 and epsin that are capable of recognizing the receptor that need to be internalized, and at the same time, they adapt the membrane and the receptors to the cluttering code. Our lab has been very interested not only in the process of endocytosis, but also on how these events affect the signaling processes. So signaling, for example, here in blue, with the blue lightning bolt pointing out to the signaling that comes from the receptor as a consequence of ligand binding and activation, but also through other signaling events, such as one that we discovered in the lab some time ago. Has to do with certain adapters like epsin interacting and affecting the activity of raw GTPase regulators, then affecting other signaling pathways, very important for cell polarity, migration, and whatnot. So these complexes are the focus of our research. And this research focus is mostly towards basic applications, but also translational applications, with a particular interest in cancer and developmental diseases. So in today's talk, I'm going to describe one of the lines of research that we carry on with cancer in our aim, and it involves the use of the ligand-receptor interaction for therapeutic purposes. So, in particularly, we are interested in strategies that change, modulate, take advantage of endocytosis for the development of anti-bladder cancer agents. So this is our major focus, although we are now branching to other types of cancers as well. So let me tell you a little bit about bladder cancer. So bladder cancer is the 4th among men and 11 among women, so you have an idea of what is the impact that it has. The first line of defense, the therapy, is the tumor resections, of course. And here we're talking about superficial bladder cancer. If it's invasive, the therapeutic approach of choice is the removal of the bladder. So tumor resection, and this is a process that is done via the urethra. So the tumor is targeted by certain endoscopic technique and is removed. Following this process, after 6 and 24 hours, there is an adjuvant therapy that is added. So there is an immuno or chemo therapeutic agent that is added again through the urethra into the lumen of the bladder. This is not a treatment that occurs via the bloodstream. It's via the urethra into the lumen of the bladder. And these agents are applied so that it can eliminate leftover cells and assure the complete elimination of the tumor. So unfortunately, these therapies are of low efficiency so far. They are also toxicity and side effects, and this is due mostly because their targeting is limited. They are also able to hit normal cells. Unfortunately, patients also develop high resistance to these treatments. So after a while, these treatments are no longer effective. So that poses a problem. In addition, some of these agents, particularly BCG, the Bacillus calmette-guerin, I will refer to it in 1 second, are in low availability nowadays. Somehow, there's been a worldwide shortage of BCG. So people are scrambling, looking for novel therapeutic approaches. And I forgot to mention, but these therapies usually involve sessions very long, 2 or 3 hours long, because of the toxicity and side effects. Patients develop sensitivity, inflammation, and whatnot, so making very difficult for them to go through such long sessions. So that's another problem with the current available therapies. All this scenario, which is less than optimal, it leads to the current status of the bladder cancer, which is to deal with a 70% recurrence post-surgery. So this is huge. There are 7 out of 10 patients may return to the office, to the clinic, with 1 or more tumors. So, and they need to start all over again the process. So as you can imagine, this is a very complex scenario. It's very taxing for the patient, but also for the U.S. healthcare system. It's considered that the cost is above $4 billion yearly. So definitely there is a need for a more efficient, with lower toxicity approach against this disease. Let me get into more details about these tumors. So these tumors arise from the epithelial layer of the bladder. So here you see in the representation the bladder on top, and a section enlarged of the epithelium that covers the bladder. The bladder lumen is where the urine is accumulated. So the lumen, the epithelia that make the lining of the bladder is made of different layers, in which the top layer is the more differentiated cells. They are called umbrella cells. And these cells secrete a glycosaminoglycan layer that is built on top of them. They establish tight junctions as well, and they secrete some proteins called uroplakins that form sort of a semi-crystalline armor that protects the cells. So all this is indeed to protect the cells, the epithelium, from the harsh environment of the urine in the bladder lumen. When a tumor arises, that's what you can see here in the blue rectangle. So the cells, the normal cells, protected by tight junctions, uroplakins, and the gag layer. But when a tumor arises, it's less differentiated. It produces very little or none of the gag layer, no tight junctions, uroplakins are not secreted, they are accumulated inside of the cells now. And so they protrude towards the bladder lumen. So this is a situation in which the bad guys, the tumors, are exposed to the lumen, while the normal cells are protected by the gag layer and the other elements I described. So it's one of those scenarios in which we could, if we add the therapeutic in the lumen of the bladder via the urethra, to be able to attack the tumor cells without affecting the normal cells. Of course, therapeutics can diffuse towards the normal cells, towards the normal cells. Angiogenesis also can be involved in the distribution of therapeutic agents in the environment, affecting normal cells. And if the therapy is effective, so it may eventually reduce the tumor mass gaining access to normal cells. So we really need not only very efficient but somehow selective agents. So years ago, we developed a strategy that we call the microclustering strategy to deliver agents into tumor cells. Why we need such strategy? As I mentioned to you, so the tumors are protruding towards the lumen, they're facing the urine. So under these conditions, the actual uptake, the endocytic event, is really poor. The cells are very low and slow for endocytosis. Each time that there is an engulfment of membrane to produce endocytosis, there is also an uptake of urine. So there is a slow rate of endocytosis for the cell. So we devised this strategy to accelerate the process. And indeed, this strategy was inspired by the mode of action of the Bacillus calmette-guerin. You remember, I referred to it, is the therapy, the adjuvant therapy of choice in bladder cancer. Here is represented by this little green oval sticking to the tumor cells. I hope you can see it. So this bacterium is able to interact with the fibronectin integrin complexes on the tumor cells, and it produces some sort of a clustering because it's multivalent. It contains many of these fibronectin attachment proteins on the surface that microcluster these receptors, and somehow the cell interprets that as a sign for endocytosis. So in the lab, we produced, in collaboration with Tim Radcliffe, an expert in bladder cancer and prostate cancer, a recombinant form of the fibronectin attachment protein of Bacillus calmette-guerin, BCG, his-tag, and we demonstrated that this protein actually is a truncation of the protein, is containing the fibronectin binding sites of this protein, could be used for the generation of strategies for delivery of therapeutics. So what we demonstrated is that if we use, for example, a polyclonal antibody against FAP, we could induce the endocytosis of this agent, of this protein. And this was because the multiple antibodies were binding to different epitopes on FAP, generating a network of antibody binding on the surface that will cluster these FAP fibronectin integrin complexes. When the cells sees this microclustering, it deduces that some complexes have been formed that need to be endocytosed, and that's a sign for the recruitment of the endocytic machinery. There are other hypotheses that we have to explain this process, which I don't have the time to discuss, but it involves bypassing certain checkpoints for endocytosis on the surface of the cell. So we adapted this technique, this process, to devise some nanoliposomal agents, nanoparticles, that will be decorated with FAP, which was his-tag, and retained in nickel NTA lipids on these nanoparticles that were spaced in such a way that will emulate the distance between the paratopes of these antibodies as is represented in the upper panel. So, in other words, these nanoliposomes present FAP in a certain density that favors the microcluster of fibronectin integrin complexes. So that's what you see in this cartoon here in the bottom. To the right, you see experiments that were done at the time with nanoliposomes labeled with CY5, with fluorescent nanoliposomes, and it allows them to detect the nanoliposomes that were taken in by MB49 cells. These are mouse bladder carcinoma cells that were expressing RAP5Q79L, which is a molecule, a GFP fusion, that it allows us to see endosomes like in large structures. So you can see in these three panels on the right On the top, the fluorescent nanoliposomes. In the middle, the fluorescent GFP labeling the endosomes. And in the bottom, the merge image that shows that some of these nanoliposomes in red are located inside of the GFP labeled structures, indicating that the uptake took place. So this was a very successful approach. We published this years ago, many years ago, almost 10 now, and was the first use of this microclustering strategy. It has been a patent, and it was this discovery of, or this design of the microcluster strategy. It gave origin to a series of generations of agents, which I'm going to describe briefly now. So the first generation is the one I just referred to, Fab-decorated nanoparticles. That was when we first used the microclustering strategy. There is a second generation that instead of using instead of using nanoparticles, it uses molecular agents. It's a single agent. It's not a particle. And those second generation agents I'm going to refer to in a second. And then we are working and developing a very successful third generation as we speak. So this second generation were molecular agents, and essentially were ligands to receptors which are upregulated in bladder cancer, like EGF, epidermal growth factor, for binding to epidermal growth factor receptor. And they were covalently fused to a bacterial toxin. In this case was the anthrax toxin. And this was a project that was carried over by Sherwin Jack, which carried most of the experiments I'm going to show, but also was taken over then by Sneha Subramanian in my lab, and currently collaborating with Andres Arias, a new student in the lab. We chose to use a fusion between EGF and this bacterial toxin because epidermal growth factor receptor is upregulated in 92% of bladder cancers. So it's a prime target for these cells. How is this working? So the fusion between EGF and the toxin is here represented by this star. I hope you can see in the white red rectangle the different elements represented in this cartoon. The star, the red star, is epidermal growth factor, and the blue oval is the protective antigen subunit of the toxin. So what you can see in the upper part is a fusion between the star and the blue oval that interact with the epidermal growth factor receptor on the plasma membrane. And as a consequence of such, the PA, the blue oval, oligomerizes, microclustering the receptors on the surface. You can see that on the right part of this upper part of the cartoon. This complex that looks like a donut is now competent for binding the second subunit of the toxin, which is the lethal factor. So in this case, it's a modified version of the lethal factor to make it even more efficient. So these green triangles representing the lethal factor interact with the donut, and then the microclustering produces the uptake of this complex, by the way I described before, and the low pH of the endosomal compartment induces a conformational change in the donut, and now it becomes a pore that goes all the way through, connecting the lumen of the endosome with the cytosol for the delivery of lethal factor in the cytosol, which it acts then in some elongation factors, blocking them and inducing apoptosis. So we tried this toxin in bladder cancer cells in comparison with several experiments, and I refer you to a paper that I will give you a reference for at the end of this talk. But we tested this agent against different other agents that are currently used for the treatment of bladder cancer, like gemcitabine and mitomycin C. And what we found is that the toxin, particularly in this case, we see the viability as a function of the concentration of the agent, what we see is that the EGF toxin, here shown in closed circles, is very efficient, has an LC50 of less than 1 nanomolar. You can see it in the dark circles, in the closed circles on the left, while gemcitabine, it has a LC50 of 80 nanomolars, and mitomycin C, an LC50 of 1,000 nanomolars on the same cells. So, as you can see, there are orders of magnitude of difference in the efficacy of these agents. So the toxin is substantially more efficient than any of these agents that are currently used in the clinic. But not only that, a very exciting, very set of exciting good news is that this toxin has the ability that other EGF-based technology has now. For example, the action of the toxin in HER2 positive cells. So HER2 is another member of the family of epidermal growth factor receptor, and is known to be counterproductive for the functioning of EGF-based technologies. And the reason is because the presence of HER2 interferes with the normal process of endocytosis of the epidermal growth factor receptor. So HER2 is capable of forming heterodimers with EGFR and interferes with the endocytosis. The endocytosis of this heterocomplex, the heterodimer, is very slow and low. So, and that is actually one of the reasons why, HER2 is such a bad prognosis marker, because it continues to be signaling from the plasma membrane. But also delivery of therapeutics via EGF-based technologies is impaired by the presence of HER2, because endocytosis is impaired. So we decided to see, because our agent was not using the classical endocytic pathway of EGFR, whether it was able to overcome this blocking by HER2. So we tried different cell lines, some of them like MB49 here on the left of this Western blot. You can see we're doing a Western blot for the detection of HER2 and tubulin as a loading control. MB49 in murine cell line does not contain HER2, but the human bladder cancer cell line T24 does, and here is a control for one that really is overexpressing a lot of HER2. And also we try samples from dog tumors. I'm going to refer to the canine model in a minute, which are actually animals that spontaneously developed tumors. And we can see that they all overexpress EGFR. Here you see 3 tumors, 1, 2, and 3. They overexpress EGFR, and simultaneously they overexpress also HER2. So we have samples with and samples without HER2 presence. And when we try the toxin, here you can see again viability, and here is depicted whether we use 1, 2, the subunits of the toxin. For example, the first control here on the left shows a negative addition of the LFN-DTA, the lethal factor, neither of the PA. Of course, this gives you 100% viability control, and the same you get if you use only one subunit of the toxin. It's not working. Both subunits are required. When you use both subunits in T24 cells, you can see that survivability drops and is capable of eliminating all the cells. Even when, as you saw in the upper panel here in this Western blot, T24 express HER2. MB49 the same, although MB49 does not express HER2, and the canine tumors also respond very efficiently to the toxin, even when they overexpress HER2, even at similar levels than EGFR. So in contrast, our approach is insensitive to the presence of HER2. So this is great news. And as I said, we speculate that it's because the mechanism of endocytosis is different. It's a microcluster based, rather than the canonical pathway of EGFR. In addition, because of that, we speculated that some mutants of EGFR, which are resilient for endocytosis, inhibited for endocytosis, will also be able to react to toxin. And that was indeed the case. We used a cell line that we had, MB49LE, means low EGFR, so it has almost doesn't express EGFR. And it's a line that we isolated in the lab. It shows the different EGFRs that we reintroduce in the cells. So we reintroduce the wild type version of EGFR, and of course, the cell viability now drops. This cell line is resistant to the toxin. So, but in the presence of EGFR, when transfected back in, is sensitive. So you can compare these two bars here on the left versus the ones on the right. On the right, we have the line that doesn't express EGFR, transfected with GFP, and it's not sensitive. It's not sensitive to the toxin. But when it's transfected with EGFR wild type, it becomes sensitive. So this is great to show the specificity of this agent towards the presence of the receptor. But also, our experiments show, listed here, which are either lacking the cytoplasmic tail, either having mutations that impair the required phosphorylation events for endocytosis, or incapable of forming dimers, can also become sensitive to the toxin. And that is because microclustering doesn't care about the canonical endocytic pathway. So another great result was that this toxin is extremely fast. Here you see cell viability as a function of time of exposure to the toxin. And you can see that even 3 minutes is enough to commit these cells to apoptosis. It doesn't mean that in 3 minutes the cells are gone. It means that in 3 minutes of exposure, it will commit the cells to undergo apoptosis, and we have seen that there's a substantial decrease of cells, 4 hours and 8 hours, with maximal disappearance of cells after 24 hours. So this is great because we can reduce the treatment time from hours. You remember I told you 2 to 3 hours patients need to endure treatment, 2 minutes with this agent. So we're very excited. We think it's a transformative approach. So all right. So another area that we tested was the ability of this toxin to eliminate cancer stem cells. Cancer stem cells are very important for recurrence and progression. You remember recurrence is one of the bigger issues in treatment for bladder cancer. So we try our toxin against different cancer stem cells. Of course, we included cancer stem cells from bladder cancer, but also from prostate, lung, and even bone marrow cancer. And we had different results. Here you see viability again versus the control and the one with the presence of the toxin. So very efficient for the elimination of cancer stem cells from bladder cancer, prostate, and lung cancer. Not so much for bone marrow cancer. And when we look at the levels of epidermal growth factor receptor, at least in these cancer stem cells, we see that the ability of the toxin to act upon of these cells is proportional to the amount of epidermal growth factor receptor. We also, in collaboration with the Department of Urology in the med school in IU, Indianapolis, we tested the toxin in freshly resected tumors from actual human patients. And I call your attention to panel C in the lower right part of this figure, in which we can see that the viability as a function of the toxin, it reaches about 85% for human samples. And in the upper two panels, you can see two pictures of two patients' cells. And what you're seeing is the epidermal growth factor receptor staining. And what we saw in patients is that, for example, patient 1 to the left, some cells express a lot, for example, that one marked with an asterisk, but the others are expressing medium amounts of epidermal growth factor receptor, and some cells do not express that much. So this poses a problem, right? Because it suggests that some cells will respond and others will not to the EGF toxin. In contrast, patient 4 is overexpressing epidermal growth factor all throughout all the cells in the tumor. So this patient is expected to respond very well to the toxin. And those results were confirmed by flow cytometry, as you can see in panel B. So this predicts that some patients will respond better than others to treatment with this EGFR toxin. So to overcome this problem, we are currently producing several new toxin derivatives with specificity for other receptors upregulated in cancer. So stay tuned. Soon it will come very, very exciting results we're getting. We tested this toxin in vivo, and I remind you, the toxin is not added in the bloodstream, it's added into the bladder via the urethra, and we tested it in normal animals, in dogs and in mice, and we saw absolutely no toxicity. And this was done by a daily observation, physical examination, urinalysis, blood count for about 2 months. I want to remind you that the toxin is so efficient that it requires nanomolar concentrations, while the intoxication dose for these toxins is in the order of micromolar. So we are definitely far from the toxicity levels of these toxins. And also, this is a binary toxin, so when the toxin, if accidentally reaches the bloodstream, it dilutes each of the components, making impossible the reconstitution of the toxin in normal cells. So the toxin, EGF toxin, is safe. So we treated this in vivo then with using dogs that are part of the program of our veterinary hospital here at Purdue that has a very successful unit dealing with transitional cell carcinomas in dogs that, as I mentioned, dogs are prone to develop bladder cancer of very similar characteristics to human ones. And so we were allowed to treat dogs that exhausted our treatment options and they didn't work, and they were overexpressing the epidermal growth factor receptor. So here I'm just showing you one example. So these are ultrasounds of the bladder prior to the treatment, after the treatment. You see, the treatment was done for between of instillation. And in average, out of 12 dogs that we have treated so far, we have seen a 30 to 40% reduction in tumor mass. This is huge because the masses of tumors are huge. Because, as I said, these animals have failed all previous treatments, and they are terminal. So the tumor mass are really, really big. So 30 to 40% is a lot of cells eliminated. And the toxin was very well tolerated. There was not toxicity associated with the treatment. The current and future direction. So we are moving towards developing other agents that will not need multiple units, but a single unit will be able to target multiple receptors and even multiple receptors of different specificity. So, so we envision that we don't need multiple units like in the toxin that they need to oligomerize, but a single agent will be able to trigger such a microcluster. And we have some prototypes that we have created for this. And by the way, these have been patented as well, just like the toxin and the microclustering technology. And preliminary results with some prototypes shows that it's very, very efficient and very fast for the uptake of nanoparticles loaded with chemotherapy. And we are expanding to other kind of cancers so far for application of this technique. So just a very brief summary, and I will end with this. The microclustering technique have been developed to induce the uptake of therapeutic by bladder cancer cells, but now we think it can be exported to other type of cancers. We have 3 generations of agents on that have been successful, and I'm very excited about these developments. And I will just acknowledge my collaborators on these projects, Dr. Tim Ratcliffe and Debbie Knapp in the Purdue Center for Cancer Research, which were very instrumental for the development of these approaches and the use in mice and in dogs. But also Drs. Michael Koch and Risto Kamakliotis in the Department of Urology of the IU School of Medicine. And in the initial phases of this project, John Collier and Brad Pentelut from Harvard and MIT contributed for the purification and production of this protein. And actually, the initial toxin was designed by John Collier. And with that, I will just acknowledge my laboratory, particularly Brian Kuhn, which was involved in the development of the microclustering technique, Sherwin Jack, that was involved in the development of the toxin approach, along now with Sneha Subramanian and Andres Arias. And of course, we acknowledge our sources of funding, particularly the National Institute of Health. And with that, thank you for your attention.